Okay, folks. So, we are still talking about communities. This is my jam. And we have talked about what a community is, right? A group of interacting species in the same place at the same time. And we've talked about how one of the critical decisions that community ecologists have to make is what is the extent of their community? What scale are they asking the question? Where are they going to stop looking at species, right? And they can do that via physical properties of a community or via biological properties. Maybe they can use taxonomic relatedness and say we're going to look at all the birds in South Florida, that bird community, or we're going to look at functional traits of an organism. We're going to look at all the nitrogen fixers in scrublands across Florida, right? And then we've talked about how community composition and community diversity are two ways that we can describe communities, right? That diversity is made up of richness and evenness, and that composition is a measure of which species are where, in addition to how many species are there, right? And so in this mini lecture, we're going to take all of that and think about ways that we can visualize community structure. So this mini lecture is about visualizing community structure. And, one of the things that I think is really important is in this class is y'all getting used to reading figures and understanding how plots of data tie to conceptual ideas. And so this chapter covers two sort of key visualizations for community structure. The first one is called a rank abundance curve. Rank abundance curve. And so what you're plotting in a rank abundance curve is you're taking all of the species in a community and you're ranking them from the most common to the least common. And then you're plotting that against the relative abundance of those species. So it's plotting the rank of the most common to least common species and plotting it against their abundances. And it looks something like this. So on the x-axis, you have species, the most common species, the second most common species, the third most common species, all the way out to the least common species. And then on the y-axis, you have proportional. Let me turn the thing so I can write it. Oops. You have proportional abundance. And so this ranges from 0 to 1, right? If it's 1, that means that species is all of the organisms in a community. If it's 0, it means that organism is none of the species in a community, right? And in your book, this is shown on a log scale, so that means that each mark is in sort of orders of magnitude. So this is 0.001 and 0.01 and 0.1 and then 1, right? And so we can build these rank abundance curves, and they can tell us or help us visualize how two species, communities of species are different. So let's imagine a couple different kinds of rank abundance curves. Let's imagine one that looks like this. Oops, let's get our red color back. Right? So we have some curve that looks like this. And then we have some curve that looks like this. Right? Let's make that line flat. I'm not the best at drawing flat lines. So take a second, pause the video, and try to describe in words how those communities differ from each other. How does the community drawn in red on the rank abundance curve differ from the community drawn in blue? So give that a pause. Take a second to look. Take a sip of my water here. Okay, so the way they differ is pretty fundamentally, right? Let's talk about the red community first. So the red community, the most abundant species, the number one. Ooh, let's make our pen red when we're writing about the red species. Does that make sense? The most abundant species in the red community is a very high proportional abundance. We're talking 0.99%, right? And then immediately that drops off to a very low percentage where the second most abundant species is, 0.001%, right? And then the third most is 0.0001, and the fourth is 0.00001 and 0.00001, right? So the community drawn in red has one very abundant species. and we would call this a long tail of low abundance species. Think about how you might describe that in terms of evenness, right? The evenness of community, the red community, is extraordinarily low. One species dominates most of the individuals, right? Alternatively, in the blue rank abundance curve, we have, the most abundant species is like 10%, and the second most is like 9%, and the third most is like 8%, and the fourth most is like 7% and 6%, and then it drops off with this tail of, less abundant lower rank species. So here we have multiple even abundance species, or nearly even. We can put a tilde to mean nearly abundant species, with some low abundance, right? Does that make sense to y'all? How that visualization really shows us the differences in community structure here. They can show us differences in evenness. So what I want to do really quickly is let me erase these and draw a couple different communities that vary in their evenness, right? And we'll use just black ink here. Just erase this and draw the whole x-axis again. We want to be in black. Okay. So we could have curves that look like this, and then they can be less steep, less steep, less steep, less steep, right? And this is increasing evenness, increasing diversity. Also, it looks like increasing richness, right? Because this one hits, 6 species, 10 species, 20 species, 40 species, right? Some of these go way further out in the rank abundance curves. Where it hits 0 is the richness, right? And so you could be asked on an exam, for example, to tell me about different rank abundance curves and how they describe different communities. So that's a rank abundance curve. That's number one. This tells us about sort of evenness, that evenness part of composition. The second kind of curve is called a species accumulation curve. And a species accumulation curve hinges on a critical idea, which is that you're less likely to find rare species, right? This makes total sense. If you go out into a community, let's imagine you go out into a tropical rainforest, an extraordinarily diverse community, and you catch 100 bugs. Do you think you're going to be anywhere close to understanding the actual richness of that rainforest? 100 individual bugs? Certainly not, right? Because there's more than 100 species. In fact, you could catch 10,000 bugs and probably not catch all of the species of insects in a tropical rainforest. We, as science, are still discovering new species as time goes on. We still haven't sampled all of the species on the planet. And because the probability of detecting, the probability of finding rare species is low, it means that the harder we look at a community, the more richness we see. And these species accumulation curves show us that phenomenon and how it varies among communities. So imagine a plot where we have observed richness here. So the observed number of species we've seen, or observed Shannon index, or whatever measure of diversity you want to use. And then on the y or on the x-axis, we have number of samples, right? Species accumulation curves have to start at the point 1, 1. You've sampled one individual and you have found one species. But then after that, they can have differing shapes. You could have one species accumulation curve that goes up very, very quickly, right? And then saturates at a very low level. So this curve has sort of 2 parameters that describe it. So I could tell you 2 things about it, and that would help you draw it. I could tell you where it saturates. We can call this cow, or something like the true richness, right? After this number of samples right here, we're not finding any new species regardless of how many samples we take. So this is probably how many species there are in that community. And then we have something that's, the rate of species accumulation. This is the other important parameter here. the slope of this, how quickly we find new species as we add new samples. Does that make sense to you? So you can imagine that a species with a higher diversity would have a higher, oops, let's undo that. We accidentally turned on our eraser. Would have a higher saturation point, right? There's more species to find. There's more species to find, right? So each of these has a higher richness because we've reached a higher asymptotic richness when we go to infinity sampling, right? But the accumulation curve, the slope of this accumulation curve, also tells us something about evenness, right? If this curve is really, really shallow, and then eventually reaches saturation up here, does that make sense to you? Actually, let's erase this whole thing and compare two curves really quick. Let's compare two curves with the same richness but different rates of accumulation. So we have a curve that looks like this and a curve that looks like this, right? So both of them reach the same asymptotic richness, right? We have the same number of species once we've fully sampled those communities. But in one of them, we get to that richness very quickly. In another, we get to that richness very slowly. Which one of those do you think has higher evenness? Take a second to imagine that. Right? So like, imagine I'm recording this near Easter, right? Imagine a bag full of jelly beans. This is a good way to think of it. And in the bag, there are 100 licorice jelly beans. I love black jelly beans. It's my favorite flavor, but I think most people don't like them. Imagine there's 100 licorice jelly beans, 1 cherry, 1 lime, 1 grape, 1 orange, right? To know how many flavors are in that bag, you'd have to pull out a lot of jelly beans, right? You'd have to pull out a lot, and you'd get licorice, and then you'd hit a cherry. And then licorice, lime. Licorice, orange. You'd have to take a lot of samples to get to the true richness of the bag. That looks like this curve, right? Now imagine if it's 20% licorice, 20% cherry, 20% lime, 20% orange, 20% lemon. You're very much more likely to get a closer estimate of that richness in fewer samples. You're going to have a much steeper accumulation curve. So these species accumulation curves can tell us a lot about community structure. They can tell us the estimate of richness, and I'll show you how we use that in real ecology in just a second. And they can tell us something about evenness, like how many samples would we have to take in this community before we actually knew how many species existed. And we haven't reached this asymptote yet for planet Earth. That's pretty cool. That's a thing you should know, right? Like we're somewhere over here for planet Earth. We haven't even documented all the species, which is wild to me. So what I want to show you actually in this lecture is really cool. So these are data from my lab, from a collaboration we've done with the University of Florida, looking at iNaturalist data, and we're looking at species accumulation curves. Well, not exactly species. I'll explain it to you. Let me show you the picture. So here's a picture. This is an accumulation curve that I made with real data from iNaturalist using annotations. So this week, I gave you all a pass on the annotations. I said, don't worry about it. I'm going to give you a 40, but read about annotations. You can annotate interactions between species. So we can go and we can see what flowers pollinators are going to. And you can imagine an accumulation curve that's something like a species accumulation curve, but instead of species on the y-axis, it's number of unique interactions. Did we see a honeybee on Abidens? Did we see honeybee on Richardia? Did we see honeybee on Hamelia? Right? Each of those is a unique interaction. And we can ask, when we look at lots and lots, thousands of iNaturalist observations, how well have we accumulated the diversity of those interactions? How close are we to knowing how many unique interactions between pollinators and plants there are, for example, in Broward County? So these are data from my lab, like I said, where we looked at a bunch of plant-pollinator interactions in Broward County and annotated them. We said, this is the animal species, this is the plant species. And it's something like, I think we're at like 15 or 1,600 observations. So the black line is the accumulation, the solid black line is the accumulation curve for interactions. And you can see that we're not even close to the asymptotic level of that curve, right? We're still accumulating lots and lots and lots of new interactions as we continue to sample this plant pollinator network. And what we can do mathematically, what community ecologists can do mathematically, is fit an equation to that line, right? Because we have some good equations that describe these accumulation curves. And that's what this dotted line is here. And decide where in the future, way out here, way out, I guess you can't see me drawing way out, but way out in the future where it reaches that asymptote. And we can ask what that asymptote is. Where does it saturate given the slope and trajectory of this line that we've already seen based on our 1,400 samples. And what we found is that there's probably something like 1,200 unique interactions in Broward County, right? So when we go into iNaturalist, we use these annotations that people like you are adding. We add some of our own annotations. We can see that maybe we've observed 500 or 600 total interactions. So 500 or 600 unique cases where a unique animal species visits a unique plant species. But using these species accumulation curves, or in this case, interaction accumulation curves, we can estimate that there's a huge amount of diversity out there that we haven't seen yet. And these are the data you're actually contributing to. So that's why I wanted to include this video in here. So these kinds of curves, whether they're rank abundance or species accumulations, there's bunches of other kinds of curves that we can make, bunches of other kinds of plots, can tell us something about community structure outside of the plain numbers, right? They can tell us something different than, here is the carrying capacity, here is the total estimated richness. You can get a lot of information out of these plots. They can tell us about richness and evenness with one line. And so that's one thing I really want you to get from this mini lecture is this sort of like visual thinking thing that's going on. I hope that makes sense. I hope these INAT data get you guys as stoked as they get me stoked. These are data that you guys are literally contributing to now that are making their way into a scientific paper as we speak. I should be working on it in addition to my lectures. So wonderful. Okay, let's keep moving. Chapter 16, communities.